Okay, so hi, I'm Hareesh. I'm working with Sitabra Sinha on neuronal networks in the field of computational. Okay, so I mainly work on the application of network science and spectral analysis in computational neuroscience. So I mainly work with this worm, the nervous system of this worm called C. elegans. It's a very ideal system to study because there are only 300 neurons, so it's computationally tractable. There are 6,000 chemical synapses, and we know very well what these neurons are, what the morphology of these neurons are, what the differentiation stages, cell lineages, positional information. We have a lot of information about the connectivity. So a natural question that people tend to ask in this field is, if you're given the structure of this particular worm, how can you infer the function from the structure? Can you say anything about the behavior of the worm just from the neuronal wiring? So some of the functions that we hope to capture are listed here. So for example, this worm has very distinct behavioral patterns of locomotion. It has very controlled propagation in the forward and backward direction. There are very distinct sinusoidal body movements. You also have these very sharp turns called omega turns, which they utilize to avoid certain harmful toxins. There is also a lot of plasticity learning based on experience, and it has the ability to sense a lot of chemicals, electric field, thermal gradients, so on. So we are turning this problem into a problem of network science. So the nodes here correspond to individual neurons, and the directed edge corresponds to chemical synapses between these neurons. So I guess we are familiar with the adjacency matrix. It's a matrix which captures the connectivity of the topology. So if the entry is 1, it corresponds to the column index and the row index, the neuron corresponding to the row and column index being connected. If it's 0, it means they're not connected. But mainly we'll be focusing on the out degree Laplacian of the network, which is basically the adjacency matrix, but the diagonal entries are the negative of the out degree of the neurons. So it's useful to think about diffusion processes when we're thinking about the Laplacian. So the decay term in the diagonal terms correspond to decay in a diffusion process, and the contribution of the neighbors is captured in the off-diagonal terms. Just to drive the point, we can actually gain a lot of information by looking at the Laplacian. So the number of 0 eigenvalues of the Laplacian tells you about the number of connected components in the network. So for example, the network shown in the corner, there are 6 isolated fragments, and if you calculate the eigenvalues of the eigenspectra of the Laplacian, you would find 6 0 eigenvalues. And the time scales associated with the eigenmodes, which describes the activity on this network, has a persistent time scale which is inversely proportional to the eigenvalue. So we can look at the smallest positive eigenvalues, and those eigenvectors have a much bigger contribution in the dynamics. So for example, if you look at something like a directed cycle, we see that the eigenmode corresponds to different waves defined over the domain of different frequencies, and the higher frequency eigenmodes, they decay much faster. They're associated with the larger eigenvalues. So this is very much in line with our understanding of diffusion process. You do a Fourier transform, you see that the higher frequency mode, they tend to decay fast. If you are given something like a linear chain, you can figure out which nodes correspond to source-like behavior and which nodes correspond to target-like behavior by looking at the principal eigenmodes and the eigenmodes that follow. So if you look at the empirical network and the Laplacian associated with it, we can look at the distribution of eigenvalues, but mainly we're interested in the dominant eigenvector, meaning the eigenvector associated with the smallest positive eigenvalue. And here I'm showing the dominant eigenvector and its components, and the color indicates the type of the neuron that is associated with each of these components. So red indicates sensory neuron, green indicates interneurons, and blue indicates motor neurons. And we can see this clear hierarchy of average activity of these components going from sensory to motor neurons. And this sort of captures this idea that sensory neurons are more like the source in these neuronal connectomes. They sense the environment, and they're mainly designed to propagate the information in a feed-forward direction. Motor neurons are more like the target neurons, and the activity pattern in the motor neurons, they will tell us something about the final behavior that you would see in the worm. So for all intents and purposes, we can think of communication between neurons as this combinatorics between the set of neurotransmitters that are released and the set of receptors that are expressed in the target neuron. So if a neuron wants to communicate to another neuron, it will release a set of neurotransmitters, and the target neuron could have some set of receptors which may or may not bind to these neurotransmitters. So you can already see the possibilities here. So if there is a node with, let's say, 5 integrase, depending on the receptor expression of the target node, you can have all possible combinations. You can selectively silence nodes that are part of the connectome otherwise, and you can have different possible configurations locally at every node. Now the question is, can we learn something from this information? So if you look at this particular plot, it's not very neat, especially between the inter and sensory neurons, because there is a lot of feedback that goes on between the network. And just from topology alone, it's very hard to distinguish which components of the network contributes to feed-forward information and which components correspond to feedback. So can we use this measure, the ratios between these average values, to selectively filter out randomized ensembles, which corresponds to information that is being driven in the feed-forward direction. So we do that, and one of the parameters that we control is the diversity of receptors expressed at every node. So in the sparse limit, we have the bare minimum number of receptors to make sure that the network is fully connected. And in the other limit, you go back to the original connectome, where you have all sorts of receptors being expressed. In the sparse limit, we selectively pick out networks which are designed for feed-forward, and we see a significant improvement in the discrepancy between the average eigenvalues of the dominant eigenvector. Now we can control the sparseness parameter, which I'm referring to as PR, and we can look at the average number of modules. So the way we estimate the module is we again use a spectral method to classify what are the modules. So modules basically mean that neurons belonging to a particular module, they're more likely to interact with each other compared to neurons that are not part of the module. So I think Rahul mentioned in one of his slides the spectral method, which is used to estimate modules. I'm using a similar method here, and what we find is for most values of PR, the number of modules and the module index are fairly consistent, except in the sparse limit. There you obtain very different modules, which is also captured by the average overlap between the modules. So we look at the sparse limit because the other limit has been fairly well explored, well studied. And what we find is, so on the x-axis I'm showing the neuron index, and on the y-axis I'm showing the module index. And what we find is, there are these very distinct modules which actually filter out some of the functionalities that are associated with the neurons in the module. So for example, the first module, it's mainly driven by interneurons and motor neurons, and most of the activity associated with these neurons are locomotion. So very distinct patterns of locomotion is encoded in this module. And there are some neurons which have been speculated to be a part of locomotion, to contribute to locomotion through different topological analysis, but there is no experimental evidence for it. And we find that they also happen to be in the same modules. We can go up different modules and we see that different sorts of sensory modules communicate with locomotion modules, motor neurons, in each of these different modules. And interestingly, we find that in 2 of these modules, some of the neurons are picked out which play a very important role in positional information during development. Now, is this a coincidence? We don't know, because there is no information about development has really gone into it. And one of the module is specifically the neurons which are designated to encode the positional information of the neurons in the nerve ring, which is basically the cluster near the brain region of the worm. And one of the modules, it very well captures certain very distinct features like withdrawal tendencies of the worm once they encounter mechanical stimulus or chemical toxins, foraging behavior. And there are some modules which are more associated with integration of information by interneurons. So the sensory neurons, they detect signals from the environment, they propagate it. The interneurons, they sort of integrate and propagate it to the motor neurons where the final activation pattern is observed. So some of these modules they specialize only in the integration part from the sensory to interneuron, like the 6th module here. So it does seem like structural analysis can reveal something about the function, but yeah, there's more work to be done. These are sort of premature results. Thanks.
Thanks, Hareesh, for this fascinating talk. Questions, please.
So I'm just curious. So these neurotransmitters. So when you have this like some input and then there have to be the molecules have to be captured. Do the molecules actually diffuse physically, or it's just a chemical process? How does it actually?
There is diffusion of neurotransmitters between the synaptic cliffs, but the length scale is so short that it's pretty fast.
I see. I see. Thank you.
Any other questions? So I had one question. So when you do this spectral analysis, you are some way like writing it in a matrix form, and something is block diagonalizable here, this matrices. So you have these modules. I can think of it as a block diagonal form of a matrix, right?
Yeah, correct.
And then what guarantees, are there any symmetries that guarantee that your matrix become block diagonal?
So there are some results which I haven't shown here. Okay. So in the sparse limit, you do see very distinct discrete eigenvalues. So you have very distinct spectral gaps when you go to the sparse limit. And there are some papers which tries to argue that if you have these sort of features, these are essentially the consequence of certain cycles and chords existing between the cycles. And there are people who try to apply permutation groups theory, weak permutation group constraints to study how these circuits are formed and stuff. So maybe it has something to do with it, but I'm not very sure about it.
Loosely, it is like permutation symmetry.
Yeah.
Okay. Any more questions? If not, let's thank Hareesh again. Yeah, thank you.